Pois.
Aí nos intervalos, você vem aqui e só fecha o microfone. Vou deixar assim.
of our afternoon section. It's a pleasure to invite the assistant professor at the microbiology department of UFMG, Dr. Monica Vieira. Sure, you can. Just turn on the. Yes.
Well, good afternoon, everyone. I'm Monica. It's a great pleasure to be here, and I thank for the organizing committee, Professor Vasco also, for the opportunity to be here talking to you today. So I'll be presenting what I've been doing for the last years concerning the interplay between leptospira and host hemostasis. It doesn't work with me.
Just a second, let's try.
You've got to be kidding.
It works.
Let's try.
Many times.
Okay, press many times. No. No. I have one.
You try, please.
No. I have one. Just a minute. Sorry for that. Okay. So, I'll start giving a brief introduction on immunity, inflammation, and hemostasis, the role of platelets in all these processes, then a bit about leptospira, leptospirosis, and then what I've been doing on the subject. So the immunity, inflammation, and hemostasis are very interconnected processes. For example, pathogens can directly activate cells of the innate immunity, which in turn activates the coagulation complement system, mediate pro-inflammatory mediators, which all activate these processes, activate themselves between two pathway interactions. All these processes allow for the activation of the active immunity also. And what's interesting is that platelets interact by many receptors and also secreted molecules with all of these processes. Well, the platelets have a primordial function, mainly in hemostasis. The primary hemostasis is mainly the formation of a platelet plug to control a tissue or a vascular endothelial injury. So when the extracellular matrix is exposed, for example, collagen and other proteins on the extracellular matrix, the platelets bind to this extracellular matrix and components and become activated. The activated platelets then change their conformation, shedding some phylopods. This is the activated state of the platelets. Then the activated platelets start to aggregate. That's one platelet bind to each other, forming then a platelet plug. That's the primary hemostasis. Then the activated platelets secrete a lot of mediators, for example, ADP, which in turn activate the secondary hemostasis pathway. That's the coagulation cascade, culminating then in the formation of fibrin. That's a mesh of insoluble protein that helps to heal this endothelial surface. Apart from the primary role in hemostasis, platelets have been each time more recognized as having other roles. That's because platelets have a lot of receptors that permit the interaction with cells from the innate and adaptive immunity, and also directly with pathogens. For example, platelets have toll-like receptors, allowing for the direct recognition of pathogens, which in turn can lead to a lot of platelet responses. They also have integrins that enable the direct interaction, for example, for the endothelial cells, E-selectin or CD62P that allow the interaction with neutrophils, which become activated and start a lot of their functions, for example, the release of neutrophil extracellular traps, and also CD40 and CD40L that are the main receptors allowing their interaction with the cells of the adaptive immunity. So we'll talk a bit about platelet-bacterial interactions. So as I told you just right now before, platelets have toll-like receptors that allow the direct recognition of pathogens. So this direct recognition of invasive pathogens can lead to direct platelet responses aiming to stop the infection. For example, platelets can entrap bacteria and also fungi in the platelet plug through the aggregation. Platelets can also internalize bacteria in a process similar to phagocytosis, and also can degranulate some bacterial, antibacterial molecules. And also because those many receptors, the platelets can, the activated platelets by bacteria, interact with the cells of the innate and adaptive immunity, leading to a lot of processes to stop the infection. For example, the interaction with neutrophils can lead to the release of neutrophil extracellular traps, lead to phagocytosis and degranulation by the neutrophils. The interaction with macrophages can stimulate the differentiation and also the phagocytosis of the pathogens. In dendritic cells, the platelets can lead to the antigen presentation. The same in B and T cells, and can also lead to the class switch of antibodies in these cells. So I think it's fair enough to say that platelets modulate a lot of functions upon an infection. And from a pathogen point of view, the interaction of platelets with pathogen can be beneficial for the dissemination and immunization of the pathogen within the host. For example, bacteria can lead to excessive stimulation of platelets, that's overactivating platelets, and lead to a consumption error, let's say. Bacteria can lead to the inhibition of the platelet function, therefore minimizing the immunomodulatory function of the platelets in the infection. And the bacteria can also cause a direct cytotoxic effect, for example, by the secretion of toxins and other components. So all of these unbalanced responses of the platelets can lead to increased dissemination of the pathogen and also immunization, making the pathogen more effective in causing an infection. And let's say that the pathogen leads to an excessive platelet activation. So as I told you, activated platelets interact directly with neutrophils, which in turn increase neutrophil extracellular traps, and also excessively activated platelets aggregate a lot. Therefore, if a pathogen excessively activate platelets, this can lead to microthrombosis within the microcirculation of the host. The excessive platelet activation can also lead to excessive endothelial activation, leading them to vasculitis with the inflammation of the endothelial vessels, the capillary vessels of the host, edema formation, and also hypotension. The excessive activation can also lead to increased release of pro-inflammatory mediators, which in turn activate B cells, T cells, neutrophils, macrophages. But then we can have a cytokine storm, for example, which can lead to high cytotoxicity within the host and in a late outcome for septic shock, for example. So these are some outcomes of the platelet-bacteria interaction, leading to a pathophysiology sign within the host. And on the other way, if the bacteria inhibits platelet activation, then the immunomodulatory functions of the platelets are, of course, inhibited, and then we have less aggregation, less coagulation, and less stimulation of the immune system. And as we have less aggregation and less coagulation, we can have hemorrhages. The same if the bacteria leads somehow to thrombocytopenia, that's the low platelet number on the circulation. Then we have less aggregation, less coagulation, and this can cause microhemorrhages to occur, and also more pronounced bleeding events. And now I'll talk about leptospira and leptospirosis. If I can manage to change this. So, leptospirosis is considered one of the main tropical infectious diseases. It affects more than 1 million persons each year in the world, especially in the tropical and equatorial regions. This accounts for approximately 60,000 deaths per year, but we believe that these numbers are very underestimated because of difficulties in the diagnosis and the case not being reported for the competent agencies. So we believe that apart from these numbers, leptospirosis also accounts for 20 to 40% of the febrile illnesses of unknown origins. So we believe that the numbers are much higher. Leptospirosis in the developed countries, North America and also Europe, is mostly related to occupational activities and also recreational activities that involve the contact with the water. In the tropical regions, it's more related to these scenarios due to the lack of sanitation, of poor sanitary conditions, and also flooding events. So affecting mainly people in low socio-economical conditions and during the rainy seasons. Those are the leptospiras. So they are spirochetes. They are coiled bacteria. At the same group, we have other bacteria of medical importance. For example, tribulina, that's important in the periodontal disease, and also causing syphilis, and Borrelia, that causes Lyme disease, recurrent fever, and leptospira causes leptospirosis. We have species living in the environment, saprophytic species, and we have species that causes leptospirosis that are pathogenic. So these bacteria are gram-negative. They are long, thin, and spiral cells, and they have two periplasmic flagella, one anchored at each of the poles of the cell. The movements of the flagella make the bacteria rotate in this rotational movement. It's very nice to see. The humans are usually accidental termination in terminal hosts in the transmission cycle of leptospirosis that mainly involve asymptomatic carriers that have mainly the kidneys colonized by the bacteria, and these asymptomatic carriers that in the urban settings are usually small rodents, rats and mice. And these asymptomatic carriers then shed live bacteria through the urine in the environment. And these bacteria are then a source for the contamination of soil, water, and other animals, which eventually lead to the contamination of humans. In humans, leptospirosis can present with a high range of symptoms, ranging from asymptomatic cases to very severe leptospirosis causing death. The most severe cases are characterized by jaundice, hypotension, hemorrhages, and multiple organ failure. Approximately 10% of the patients die from leptospirosis. Although the importance of leptospira both in the humans, clinic, and also veterinary, the molecular mechanisms of the pathogenesis and virulence are poorly known, almost unknown, I would say. We don't know most of the virulence factors involved in leptospirosis, and the origin of the symptomatology and pathophysiology of leptospirosis are almost unknown. We don't know which factors interact with the host to cause the symptoms that we see in the patients. Apparently, leptospirosis don't have toxins or classical virulence factors that justify the severeness. Is this correct? I don't know. Yes. The severeness of the disease. What we believe is that the excessive immune and inflammatory responses which lead to the hemostatic imbalance might be involved in the leptospirosis pathogenesis. And that's supported by some evidences, some symptoms presented by the patients. For example, fever, hypotension, high levels of cytokines, massive infiltration of immune cells in the affected organs, for example, liver and kidneys, vasculitis, so the whole circulatory system of the host is compromised. Thrombocytopenia is a very important effect and is associated with a poor outcome of the patients, and also the occurrence of hemorrhages. And one type of clinical presentation, that's the pulmonary hemorrhage, is becoming each time more common in leptospirosis and leading to almost 50% of death of the patients. And nobody knows why it's becoming more common and which factors of the leptospirosis lead to this outcome. Now a bit of host-pathogen interactions in leptospirosis. So I've been working with leptospira and leptospirosis since many years, since my initiation scientific, and I've been involved in several projects. First, we showed that leptospirosis interacts with the fibrinolytic system by binding plasminogen, that's the key zymogen circulating in blood, which is then activated to plasmin, which then cleaves fibrin that's produced during the coagulation. So leptospirosis bind to plasminogen through various receptors on the surface. Leptospirus also stimulates the secretion and production of plasminogen activators by endothelial cells, and these free activators then activate the bound plasminogen to plasmin. Then bacteria are equipped with a broad spectrum serine proteins on the surface, which then aids the bacteria to disseminate by transpassing the endothelial monolayers, and also leading to the degradation, for example, of opsonizing antibodies and complement system molecules on the surface of the bacteria. So we have shown that the interaction of leptospira with the fibrinolytic system in vitro can lead to increased penetration and immune evasion, and in vivo we have the evidences that the fibrinolysis is activated during leptospirosis because patients present increased levels of the plasminogen activators, both the ureakinase type and tissue type, and they have increased levels of fibrinolysis markers. We have shown also that leptospira interact with, by many ways with the coagulation system. For example, binding to factor 12 on the surface, and also high molecular weight kininogen. Then the prekallikrein activates all the contact system, leading then to the release of the pro-inflammatory and endothelial active molecule bradykinin. We have also shown that the activation of the contact system on the surface of leptospirus leads to the activation of the intrinsic pathway of coagulation, and also that leptospirus activates the extrinsic pathway of coagulation by stimulating the production of tissue factor from monocytes. We have also shown that patients have a high number of microvesicles shed by monocytes bearing tissue factor, therefore leading to a procoagulant state on the host. We have also shown that leptospirus allow the formation of the prothrombinase complex on the surface, which then favors the production of thrombin and then activating all the coagulation cascades. And that's a summary of how bacteria can interact with the coagulation system. So in vitro, we have shown that leptospira stimulates the coagulation and also inflammation by activating this inflammatory branch of the coagulation pathway. And in patients, we have shown that they have increased markers of activated coagulation and also increased markers of activated inflammation, for example, higher levels of bradykinin. And we have shown that leptospirus interact directly with neutrophils, activating them to release HBP, or heparin binding protein. That's a protein that increases the vascular permeability and increasing the vascular leakage, then possibly leading to formation of edema and hypertension. And we have also shown that leptospira stimulates the production and activation of pro-metalloproteases, which help to increase the vascular permeability and increase leptospirosis dissemination within the host. So in vitro, we have shown the increased penetration and increased permeability as a consequence of HBP and metalloproteases secretion and activation. And in patients, we have found that they have a very high level of HBP. We are also proposing this as an early marker of leptospirosis infection. They have increased levels of bradykinin, as I have said before, and increased levels of metalloproteases. And with all this in mind, and knowing that platelets interact in all of these processes, now I'm more interested in understanding if leptospirosis interact with platelets and the outcomes of this for the infection. So I want to understand the molecular mechanism of interaction between platelets and leptospirosis and the consequences both for the host and the pathogen. How this can modulate the host responses, thinking in immune, inflammatory, and hemostatic processes. Also, how this contributes to leptospirosis pathophysiology. For example, the occurrence of hemorrhages and thrombocytopenia, and I hope to propose novel strategies against the disease, both therapeutic, prophylactic, and diagnostic. So I want to understand if the platelets interact with leptospira can lead to antibacterial response, or if this leads to a pro-thrombotic state within the host, how platelets interact with the immune system by secretion of pro-inflammatory mediators, and also modulating the response of other immune cells, and also understand the activation state of platelets both in vitro, in animal models, and in vivo in patients. And also if this interaction can lead to platelet-leukocyte interactions. And I would say that's my main focus, is to understand how leptospira leads to thrombocytopenia. And we have four main hypotheses. That's the decrease in the production of platelets, increased clearance, increased cytotoxicity of bacteria in the platelets, and also maybe a consumption process. That's the increased activation, overactivation, and also by hemorrhages and disseminated intravascular coagulation. Most of the pathogens stimulate platelets. And surprisingly, when we put leptospira and platelets together, nothing happens. So I propose this project believing that platelets will overactivate platelets and we will have very nice results. But no, nothing happens. So leptospira seems to be unable to directly activate platelets and lead to platelet aggregation. And that's very uncommon for pathogens. We also measured the platelet activation by surface markers. So we have here the positive control, that's ATP, that's a non-stimulator of platelets, which is very nicely is expressed here in the surface. And leptospiras make nothing. We have also a marker of dense granules and lysosomal content secretion, and we have no response. And also when leptospiras are put together with platelets, we don't see the activation of the GP2B3A integrin. That's very important for the interaction with the endothelial cells. And we have also shown by morphological changes in secretion of granules that leptospiras do not activate platelets. And also we have shown that leptospiras seem to detach pre-adhered platelets. So we adhered platelets on glass surfaces and then incubated with platelets. When we treated only with PBS, nothing happened. When we treated with ATP, we had platelet aggregation, as expected. And when we treated with leptospiras, we had the release of platelets from the glass surface. So something was happening here. And we did this with platelet-rich plasma and also with washed platelets. And we had the same results. And when we use the washed platelets, meaning that we don't have the plasma components, we saw a very marked damage on the platelets. So we could see small fragments of platelets adhered to the surface of the glass cover slip. And here in more detail, the effect of the leptospiras on the platelets then leading to the damage of the cells. Some cases we had what appeared to be a perforation of the platelets, and we could see very nicely the direct interaction of leptospiras with the platelets. Here by transmission microscopy, we could also see that we have direct interactions. Here is a leptospira, also here the arrows show the leptospiras directly interacting with platelets. And while we have the morphological changes of platelets with a lot of halopodia and intense degranulation when we treat with ATP, we have morphology compatible with unstimulated platelet, round cells with all the granules inside the cytoplasm. And very interesting, in some cases we observed the leptospiras inside the platelets. We still don't know if this is an active process of either leptospiras entering platelets or platelets internalizing leptospiras. We still don't know. And we also saw events of microvascularization of platelets, apparently with different compositions. analyzing the inside of these microvesicles. We still also don't know the meaning of this microvesiculation, the composition of the interior of this microvesicle. It's just an observational analysis. And interestingly, we also have shown that leptospiras do not lead to the loss of viability of the platelets, so they seem to be perfectly viable after the interaction. So what we propose is that, for example, while ADP, that's a non-platelet activator, lead to an activated state, procoagulant, apoptotic platelets, via mechanisms dependent of activation and degranulation, via the increase of intracellular calcium and mitochondrial membrane depolarization, leading them to a very high aggregative phenotype. When we incubate resting platelets with leptospiras, we see a phenotype that seems to be independent of activation and degranulation, which leads to platelets not able to aggregate, but they seem to lose the ability to adhere to surfaces and release microvesicles. And we still don't know if this can lead to increased clearance of the platelets by this kind of phenotype. Do I still have time? And I've been working also with some recombinant proteins that through bioinformatic mining on the genome of leptospira interrogans, I found some interesting proteins that might be participating in the imbalance of the hemostatic system in leptospirosis. So, for example, I will show you some results of the proteins Bact-A and Bact-B. Bact not for bats, but for Bacteroides aerotolerance proteins. So we found that these proteins contain von Willebrand factor type A domains. So the von Willebrand factor is a physiologic molecule circulating on the plasma, and it is a carrier for the coagulation factor 8. And it's known that the von Willebrand factor mediates the platelet vascular endothelial adhesion. So, and this property of adherence to the endothelial cells, the vascular endothelial cells, it is mediated by the type A domains. So that's why I was very interested to study these proteins. And it's also known that the deficiency in the von Willebrand factor, the physiological von Willebrand factor, leads to hemorrhagia. So something nice could arise from the analysis of these proteins. So we produced the recombinant Bact-A and Bact-B. Nothing to say here. And these proteins, both Bact-A and Bact-B, when we incubated with purified fibrinogen, we have saw that they degrade the fibrinogen. The effect of Bact-B was more pronounced, generating more bands. but Bact also promoted some fibrinogen degradation. We also have shown that especially Bact can also cleave other substrates, for example, gelatin, here also the fibrinogen, and casein. These are zymography assays. And when we put together recombinant Bact and Bact with platelets and analyzed the degradation, we saw, for example, that we had no effect when we added ATP, collagen, or epinephrine that are known as platelet stimulators, activators. But when we stimulated the platelets with thrombin, both in platelet-rich plasma and in washed platelets, we saw a marked reduction on the platelet aggregation. And also when we add ristocetin, that's an antibiotic that promotes the binding of von Willebrand factor to platelets. We also saw a reduction of the platelet aggregation. So we believe that these proteins might have some effect during the infection. And how our hypothesis is that these proteins might inhibit platelet adhesion and activation by competing with the receptor of thrombin and von Willebrand factor. That's the same receptor, the GP1B. And we believe that this might have also an impact on the occurrence of hemorrhages, as this protein degrades fibrinogen, that's important for the secondary hemostasis, the coagulation cascade. And by inhibiting platelet aggregation, here by inhibiting by competition of the binding site of the receptor. So we believe that these effects might lead to the inactivation and also might promote the increased platelet clearance within the host. So that's what I wanted to show you today. And I'm open for questions. And thank you for the attention.
Any questions?
Thank you so much for the presentation. I was wondering if those platelets pre-stimulated with leptospira could stimulate neutrophils in the same way, even if they don't have any morphological changes. Did you see anything about that?
We still did not test this, but it's interesting, yes.
Another point, how about the in vivo infection? Do you have animal models for these, and maybe to understand the relationship between neutrophils or whatever, the interaction with platelets, activate or not?
Yes, that's something that we want to do, of course, to test in animals, but as you might know, the animal model for leptospirosis are hamsters, and that's not very easy to do this here. I'm more or less new at UFMG. I arrived just before the COVID pandemic, so my research was very my research was stopped during this whole time, and now I'm returning the bench experiments, and hopefully I will start to see where can I find hamsters to start to work with. But of course, it's one of the the next steps.
Thank you so much, Professor Monica. Earlier in your talk, you mentioned that not many viral infectors from leptospira are associated with leptospirosis. Is that due to poor knowledge of the viral infectors, or because there are no related interactions between the viral infectors and host molecules?
We believe that leptospiras have unique virulence mechanisms. If we look at the genome of this bacteria, we will see that more than 50% of the proteins are hypothetical proteins, not related with anything known. So we believe that it's a very unique pathogenic mechanism. And we have some proteins, the ones that work with leptospira, of course, we know EPL32, EPL45, and super expressed proteins within the cell that were the golden targets for the leptospirologists. But still today, after 15 years, I don't know how many years, we still don't know what these proteins that are super expressed do. We cannot see a correlation with virulence, so we don't know the virulence factors. We know that LPS is important, and that's almost everything that we know. So that's a lot of work to do.
Any other questions? So I just received a question from Larissa, a PhD student. She's asking about the vesicles from the platelets autolysis. Does it occur during leptospiria? I don't know how to pronounce the word, sorry. Is there anything that indicates these vesicles would be a marker from clinical diagnosis?
I've seen these microvesicles from the platelets only in vitro by now. I have not investigated in vivo in animal models or in patients, so I don't know if this occurs in vivo, but if it occurs in vivo, of course, there will be implications for pathogenesis.
Anyone else? So thank you, Dr.
Thank you.
Following on, we are pleased to have a senior researcher at Inflam, leader of the microbiology team, Dr. Higueton.
Just a precision, I'm not a senior. Someone. No, sorry. I don't know. You know, I'm a senior. Technology is not bad. Yeah. Go on here. Go back here. So good afternoon, everyone. First of all, I would like to thank Vasco, Flavia, the Veterinary School, and the UFMG to host us and for the organization of this closing seminar of the LIA project. I hope that it will continue in the future, and we have a second version of this LIA. So today, I would like to talk about, my name is Eric Guénon, I'm a senior scientist, as you know, and I'm coming from INRAE and Rennes, a very beautiful city. So today, I would like to talk about bacterial extracellular vesicles, and what I would like to do is to give you an overview of what you know about these vesicles. And for this, I would like to use data that we produce during the LEA project with our students that we have between UFMG and Inflam. So let's go for the trip. So what are extracellular vesicles? Do you hear me? Yeah, it's not so okay. Perfect. So what are extracellular vesicles or EVs for the next part of the talk? So EVs are lipid bilayer spherical nanocyte particles ranging from 30 to 300 nanometer. In general, the average size is 100 nanometer. They are produced by most cells in all domains of life, from eukaryote, prokaryote, gram-plus, gram-negative bacterium, RT, and are secreted in the extracellular environment. They carry various molecules, such as proteins, lipids, nucleic acids, that originate from the parent cells, and they can transport and deliver this molecule to local or distant cells to interact and modify them. They are now considered as conceptual mechanism for intercellular communication and inter-kingdom communication. So here on the left side, is it the left side? The left side of the slide, different pictures showing extracellular vesicles from transmission electron microscopy. We have a bacterial cell and extracellular and EVs here. From cell, again, the cell and the EVs, and after their purification from the future supernatants by EM, we can see EVs here, and by cryo-EM, we can have EVs with the spherical shape. So the discovery of EVs is that back on 80s, yeah, from a gram-negative bacterium, E. coli, and the first description of EVs from a gram-positive bacterium is approximately 10 years ago in Staphylococcus aureus. And between these two periods, we have different discovery, mostly in eukaryotic cells, on the biogenesis and the functions of this EV. This is illustrated here. This is the number of publications per year, and we can observe in 10 years ago a burst of the number of publications on EVs. And if you look on this publication, most in black concern eukaryotic, while the white bar are very fine. And when we look more precisely on this white bar, we can see that most of the discovery of the description on EVs were for gram-negative bacteria, and not very few publication on gram-positive. So it's really an essential field of research for gram-positive bacteria, and also the growing interest for EVs is driven in large part by their potential application. Indeed, it's proposed that these EVs can be used as circulating biomarkers due to their property. For example, they are involved in multiple pathology. They are both specific to the cell type, are present in almost all body fluids, and are stable. And like this, it has been proposed that they can be used for diagnostic, prognostic, or evolution marker. The second application with these EVs is the use of them as delivery system. Again, due to their properties, they can be used as therapeutic delivery tools for disease treatment, immunotherapy, generative medicine, and vaccine delivery. So this is not the aim of my talk today. And today, what I would like to share with you is what we know about EVs from bacteria, and in particular in terms of their biogenesis, their cargo, their mechanisms of action, their functions, and where we can find these EVs. So for that, I mentioned earlier, I will give you some example from our lab on two types of producing cells. The first one is Staphylococcus aureus, a gram-positive bacterium and a pathogen from human and animal. And the second one is a Propionibacterium frodenreichi, also a positive gram-positive bacterium, which is a bacterium of food interest with probiotic effect. And what we particularly study in the HCLO is the biogenesis of these EVs from these two bacteria, their cargo, their mechanism of action, and their function in relation with the host cells. So, in terms of mechanism, just to precise some words, probably you know the name, the word exosome or microvesicule and so on. So there is a lot of different words to mention extracellular vesicle, which is a generic term. In eukaryotic cells, we have mainly two types of extracellular vesicles. The first one is the exosome, and these exosomes are produced from the endosomal pathway. And the second one, larger particule, compared to the exosome, microvesicule, are produced by the budding of the plasma membrane of the cell. Whatever the pathway, there is no real differences between their contents. It's not totally true, but for bacteria, gram-negative bacteria, extracellular vesicle are also known as other membrane vesicle, or OMV, because they are produced from the budding of the other membrane of the gram-negative bacterium. In gram-positive bacterium, they are formed from the budding of the cell membrane, and in this case, we call this particule as a membrane vesicule or extracellular vesicles. And finally, there is another way to produce vesicle from gram-negative and gram-positive bacteria if it's after cell lysis and bleeding of the cell membrane. Okay, in terms of mechanistic of the production of these particles, clearly it's the vesiculation is a complex and still elusive process. We don't know a lot of things about the mechanism involved in their formation. What we know, what is proposed in gram-negative bacteria, is that these particles can be formed in particular region of between the other membrane and the cell membrane. For example, in region in which we have an accumulation of proteins or peptidoglycan and so on, so different molecules that create an invagination, or not but an exvagination of the other membrane, and by the tubular pressure, we have the creation of the liberation of the vesicle. We have another model in which this region is a loose region between the two, and the last is a region in which we have a microdomain lipid with different charge, and they can be produced like this. But again, we don't know exactly how it works. In gram-positive bacteria, similar model has been already proposed for their formation, but one difficulty, additional difficulty for the formation of EVs is to escape the cell wall, which is a thick layer between the extracellular environment and the cell membrane. And in this case, it has been proposed that some proteases can integrate this cell wall to liberate the extracellular vesicle, or also the presence of particular channels that can be used by the EV for their secretion. but I don't know that this channel exists. We have no data from that, for that, sorry. But what we know is in Staphylococcus aureus, we have a particular model. It has been shown that small peptides, PSM peptides, can cut the cell membrane, and this cut can promote the formation of EVs, and autolyzing will degrade the peptidoglycan to liberate this particle. But it's a specific model of staph because PSM is found only in staph aureus, and autolysins are very particular also of this species. So again, for gram-positive bacteria, we don't know a lot of things about the process of their formation. What we know is that if we perturb the lipid homeostasis, the peptidoglycan homeostasis, the cell envelope, or if we induce the phage, we have the formation of EVs. And for that, different conditions can be trigger this process. Iron limitation, antibiotic, enzyme that degrade peptidoglycan, signaling molecules, the hydrophobic compounds, the DNA imaging agents, and so on. So there is different things that can trigger their formation. In terms of environment, we can find EVs in almost all environments, since they are secreted extracellularly. We found EVs in the culture spermatens of bacteria, in culture medium of eukaryotic cells, in biofilm, like this, in intercellular space of tissue, in human or animal, and other eukaryotes, and almost all body fluids. So extracellular vesicles are everywhere. In terms of their cargo, super. Oops. Yeah. Okay. In terms of cargo, the cargo of EVs is this is the cell membrane here, and this is the representation of an EV's information. And the cargo is really loaded during the vesiculation process. And this cargo, as I mentioned previously, include proteins, lipoproteins, DNA, RNA, metabolite, and so on. And what I would like to show you now is the cargo of Staphylococcus aureus and Propionibacterium fraudenreichi. And we have looked on their protein contents and RNA contents, and also for Propionibacterium fraudenreichi on their protein contents. So this is the work of Brenda, and I invite you to assist to the defense of Brenda in the next Friday to have a better information of this. But what we know about the content of EVs from that is in terms of RNA. We can identify all class of RNA. It means mRNA, tRNA, RNA, small RNA with regulatory function. Most of these RNA are fragmented, not degraded, but fragmented, and approximately only 5% of the RNA identified in EVs from staph are intact. And also, they contain very long transcripts. We can identify transcript over, I don't remember exactly, but 10K. So we have a very long transcript and intact transcript. So this may raise the question about if these RNA are functional and their biological role. I'm very sorry. In terms of protein content, we perform this analysis in EVs from staph from clinical isolate, bovine isolate, but also in Bacterium Flavonoria PF. And okay, whatever the strain or the species that produce the EVs, what we observe that we can also find in the EVs all the type of protein. I mean, all the type in terms of localization. We have protein with that localize in the cytoplasm, in the membrane, or extracellular protein. We can also identify protein with lipo signal or sex signal. In terms of function, also, we can have, I don't know, but protein associated with metabolism, with transcription, translation, and so on. So we can have all the type of protein in these EVs. And but this protein or their content reflect the status of the parent cell. In particular, they are specific of the cell type or of the strain. For example, here we characterize the proteome of EVs from bovine strain of staph, but also from a human strain. And what we can observe is that we have a core proteome, but also specific proteins that can be formed specifically according to the strain. When we analyze these different proteins and we make a clustering of them, we can, I don't know if you see something else, well, anyway. When we clusterize this data on the presence absence of this protein in the EVs, we can cluster the EVs according to the origin of the strain. For example, here we have bovine strain, here we have ovine strain, and here we have the human strain. When we make the same experiments, but not with the presence absence, but based on the abundance of the protein inside the EVs, so we are looking just the core protein here, and we clustering the protein according to their abundance. In this case, we have a clustering of the EVs, not on the origin of the EVs, but on the severity that the strain can induce in their host. It means that here we have mild strain, and here we have strain that are able to induce severe infection. So, and we also are able to identify the protein that are responsible of this clustering between EVs. So again, we have the cargo of the EVs reflects the strain properties. This cargo is also specific of the physiological state of the cell. For example, here we analyze the proteome of EVs from PF cultivated in two different conditions. In one medium called UF, and the second one in medium called yield. We have similar protein, but also different sets of proteins that are specific of this media. And when we compare again the abundance of the proteins, we have also some proteins more abundant in EVs coming from this condition compared to this one, and that is also true. So their cargo depends on the growth condition at the level of their quality and quantity also. And we have the same results for the RNA cargo of the EVs. So it means that the environmental condition can modulate the EV's properties. And this is true for their physical characteristics, for example, in terms of their size, but also in terms of quantity, rate, in terms of cargo, but also in terms of their activity. And according to the growth condition, we can have EVs with different properties in terms of activity. So it means that we can modify or engineer these EVs. And this is what we observe, but we have similar conclusion from the literature in different other bacteria. So finally, what we also observe is that the EV cargo is selectively loaded into the EVs. Indeed, when we have a look on the composition of the EVs, it's the protein composition at the stage of the EVs here, the pattern is very different from the pattern that we can observe from the whole cell. And indeed, by different tests and statistical studies, we can observe that EVs are enriched in a certain category of proteins. For example, here in lipoprotein here, and membrane protein. It's true for their localization, but also for their properties. For example, here we have an enrichment for the protein involved in the ribosome and translation in EVs when compared to the whole cell. So, and again, what we also observe is that for staph, we have 10% of the protein in EVs that are associated with the virulence, either virulence factor directly or the factor associated to the infection. So it seems that selective or specific mechanism for the packaging of the proteins may exist. And for the RNA cargo, this is the work of Brenda, we have similar results. We have some specific mechanism that drive the content of the EVs. So with this result, we're wondering whether if we can analyze more precisely the rules involved in this sorting. For that, we use a robust set of protein, the cargo here, the core protein, that we built with the comparison of these different EVs, and by a different approach, we try to find some rules. The first one is to find if we have some specific signal or motif on the proteins, on the sequence of the proteins of these present in these EVs, and we detect no particular motif. But what we observe is that in EVs, protein are more positively charged in EVs when compared to the whole cell proteome. We have more lysine in this protein, and also this protein have a codon adaptation index higher than the rest of the protein. So here, this is a PCA, so a principal component analysis of the proteome and we can see here the EV's protein, and in blue the whole cell protein. And it's difficult to separate the protein present in the EVs, but we have a tendency, and here we have indeed positively charged protein, with a protein with a CA higher, and also content higher in lysine. So it seems that these properties seems to be have a positive role in the sorting of the protein for the EVs. And this is another example in propionic Freudenreiche EVs. And here we use a different approach by intelligence artificial. This is the work of Vinicius, And we arrived to the same conclusion with more protein with lysine, positively charged protein in EVs when compared to the rest of protein. So it seems that some rules exist to explain why a protein is present or not in EVs. Okay, so just to conclude on this part, we need to have a better, I think, biophysical characterization of EVs, notably in terms of charge, elasticity, and resistance, because for the moment, only what we do is just to measure their size and the quantity. But I think that also the charge of EVs and resistance should have an impact. Again, when we need to have some better characterization of smaller RNA present in EVs, notably in relation to the infection and the interaction with the host cells. As mentioned, the impact of full-length transcript and fragmented RNA in EV need to be assessed. The lipid composition and the presence of metabolites in EV is very scarce. So again, I think that we need to improve this knowledge. The mechanisms of protein cargo sorting is far away to be solved. So we have a lot of work to do. And the genetic and complemental modulation of EV and production and composition should also to be assessed. So in terms of mechanism of action, whatever the EVs from eukaryotic or bacteria, so microvesicle, exosome, OMV or NV, there are three different ways for an EV to interact with recipient cells, and particularly eukaryotic cells. The first one involves what we call the receptor ligand interaction, so an interaction at the cell surface of the EVs and the host cell that can be involve sugar or protein interaction, and then signaling. And this is what we know about the EVs of Staphylococcus aureus, that is able to interact with TLR2, the toll-like receptor 2, with specific FTA lipoprotein, specific lipoprotein. The second one is the direct fusion of the EV membrane with the EV of host cell and the delivery of its cargo that can modify the host cell. And finally, by phagocytosis or endocytosis. And in this case, there are different ways of endocytosis. For example, micropinocytosis, phagocytosis, lipid graft, caveolin-dependent endocytosis, or clathrin-dependent endocytosis. So many ways. For staph, the EVs produced by staph, we decide to analyze the interaction with the interaction of the EVs with the host cell, and for that we labeled with a specific fluorescent dye the EVs, and we incubate the EVs with host cells, and we measure the transfer of the fluorescence from the EV to the host cells. And we perform these experiments with different concentration of EVs. And what we can observe that in function of the EV concentration, we have an increase of the, it's better we have an increase of the fluorescence to the host cells. We also perform this experiment in function of time, and we incubate 20 micrograms of EVs with host cells, and for 30, 1 hour, 2 hours, and up to 70 hours. And what we observe also is an increase of the fluorescence to the host cells. And we interpret this increase of the fluorescence by the internalization of the EVs into the host cells. And this is dose dependent and is time dependent. And to confirm these experiments or these results, we use confocal microscopy in the same condition, and we label EVs with a red dye and host cell with 2 dyes, the blue for the nucleus and the green for the actin. And what we can observe is the presence, of EVs in red inside the host cells. So we have a 2D picture of this interaction and localization of the EVs in the cells. And this is true at 3 hours post-incubation, and we have a lot of internalization after 70 hours. As you can see, we have more red dots in the cell. So to go further, we use a specific inhibitor that block some pathway, for example, for the internalization of the EVs. For example, the dinosaur, which block the clathrin-dependent endocytosis, or amyloid that blocks the micropinocytosis here. And we perform the similar experiments in flow cytometry or by confocal microscopy. So when we, this is the control panel with just the incubation of EVs with host cell during 3 hours, and we can observe the EVs inside the cell. In presence of chlorpromazine, which block the clathrin-dependent endocytosis, we have no real differences between the two. But when we use a dinosaur of amyloid, we cannot observe EVs inside the cell. So it means that in presence of these two inhibitors, we block the entry of the EVs in the cell, and it means that EVs can be internalized by a mechanism using micropinocytose or dynamine-mediated endocytosis. So this is true for the EVs we use from this strain, but if you use another strain, you can have another results. For example, with this one, this work showed that it's only via the dynamine that the EVs enter into the cell. So several pathways can be used by EVs to interact with host cells, and the mechanism of their uptake could be strain dependent and cell type dependent also, of course. So now we move on their function. 2 types of functions have been described for bacterial EVs. The first one is defensive function. So this is the EVs can, for example, share antibiotic resistance in the population to increase their resistance. Can enable some biofilm formation, mechanism of cross-feeding to improve the population, transfer of genetic material, and so on. And also offensive function, for example, to kill competing bacteria, to deliver toxin and virulence factor to host cells, to disrupt tissue, and to elicit inflammatory response. So in our lab, as you know now, we are interested by 2 bacteria, P. fraudenreichi and Staph aureus. One is a probiotic, and we are interested in its property related to their ability to immunomodulate the host response, and Staph, the pathogen, for this ability to be a virulent agent. And so we analyze the EVs produced by these two organisms related to this property. Okay, this is what we observe from P. Foldenreichi when we analyze the proteome. So when we analyze the proteome, we observe that inside the EV or associated to the EV, we can identify different proteins involved in the immunomodulatory functions of the producing bacteria. And Vinicius decided to use a PPI strategy to identify the potential targets of this protein in humans. And he observed that NF-κB was another with a lot of interaction with the protein found into the EV produced by this strain, suggesting that EVs from PF can have a role on the immunomodulation. In particular, we found that several proteins such as SFPB, which is known to be involved in the immunomodulatory function of PF, can be related or interact with NF-κB. So this is our hypothesis, and for that, to demonstrate this hypothesis, Vinicius measured the liberation of IL-8 from HT29 cells in presence or absence of EVs. So in absence of EVs is the control, the TBS, in presence of EVs, and we have no differences when the cells were untreated. But when we treat the cell with LPS, so we have an induction of the IL-8 production. Indeed, LPS is a pro-inflammatory inducer. And when we incubate the cells treated with the LPS with different concentration of EVs, we have a reduction of the inflammatory, in the reduction of the IL-8 production which is similar to the basal level here. And so we observe a modulation of IL-8, which is dose dependent. And we performed similar experiments not on the IL-8 production, but on the regulatory activity of the NF-κB regulator in the same condition and in the HT29 cell line also. And we observe exactly the same thing. So in absence of LPS, we have no effect of the EVs. In presence of LPS, we have an induction of the regulatory activity of the NF-κB. And when we apply EVs, we have a reduction of this activity. So a reduction of the inflammation or the activity of the regulators. So this is the NF-κB pathway, and we induce the pathway here with the LPS. But as you can see, there are many ways to induce this pathway. NF-κB is here. There are many ways for that. And we decide to better understand how these EVs can modulate this pathway, to use different inductors. But when we use TNF to induce the cell, and when we apply EVs, there is no variation of the production of IL-8 or the NF-κB activity. And we have the similar for the IL-1 beta. When we induce the cell with this inductor, and when we apply EVs, there is no difference in the NF-κB activity and the IL-8 release. So it means that this response is really specific to the LPS induction. And it means that EVs probably can act here, outside, in competing with the LPS for the TLR4 or inside the cell to inhibit some specific part of this pathway. And we don't have the answer to this, but we use SLPB mutant, so a mutant for a protein involved in the immunomodulatory properties of the bacteria. And we purify the EV and compare the EV of the mutant and the wild type strain. And as you can see here, the wild type EVs can reduce the NF-kappa B activation. And for the SLPB mutant, we have a reduction, but it's just partial. So it means that this protein is probably involved in this immunomodulation, but we don't know if it's directly or indirectly, and SLPB is not alone. And when we treat the EVs with protease K to degrade all the protein at the EV surface, we have no effect. So it means that if SLPB have an effect, it's not outside, probably after the internalization of the EVs in the cell. So this work is ongoing, and what we want to do now is better to understand the mechanism of action of these EVs. So the mode of action, how the EVs are internalized, identification, we want to identify the bacterial effector, the cellular target, the effect of the EVs also on and compare the effect of the EVs and the bacterium, and so on. We want also, if it's possible, to evaluate the effect of the EVs in vivo, which is not so easy to do. The role of this EV in other cell models and other contexts, of course, and to see if EVs produced by this bacterium, can have a role in the communication with other bacterial cells, and evaluate also the intraspecific diversity of EVs of propionate. So now for the last part, some data about the function of the EVs produced by Staphylococcus aureus and related to the pathogenesis, of course. So this is we talked about this morning, and we demonstrated that EVs can carry both factors involved in the colonization of the host, but also in the severity of the infection. And again, I show you that 10% of the protein present in the EVs are related to the virulence. So probably EVs are involved in the virulence. And we demonstrated that EVs are not cytotoxic, but are able to modulate the host response in vitro. And we compare the response, it's the IL-8, I don't see it, IL-1 beta E, TNF alpha, so the induction of different cytokines, and in presence of the bacteria, the ATA, the bacteria but kill, and different concentration of EVs. And what we can observe is that EVs are able to induce the expression of these genes, and sometimes in the same way, with the same ratio than the live bacteria. Brenda also performed a transcriptomic analysis, and we observed that we have a lot of genes modulated by the presence of EVs, depending on the time of the incubation, from 50 to 200 genes of the eukaryotic cells can be modulated by these EVs. And when we compare this response with the one obtained with the bacteria, we have some common genes, but also specific genes. It means that probably the EVs are able to induce different genes than in bacteria. And okay, and this is an example here in terms of inflammation and immunomodulation. But what we also observe is that other pathways, from example ribosome, DNA replication, metabolism, can be also induced or modified by the EVs of staph. So the action of the action of EV is not restricted to the immunomodulation, but can be larger. We perform an in vivo experiments in a mice model in which we inject in the mammary gland of lactating female either PBS, which is the positive and the control, sorry, the bacteria, the living bacteria, the inky bacteria, and the LTA, which is a positive control, and two different doses of EVs. And we analyze 24 hours after the injection, the effect, the impact of this different injection after the sacrifice of animals. And this is the mammary gland of the healthy animal, so everything is clear. This is the mammary gland of the animal treated with the living bacterial cells, and you can observe some lesion here with hemorrhagia, edema, and so on. With the it-kill bacteria, we have also some lesion, but in this case it's moderate when we compare to the live bacteria. And when we use EVs, we have also a moderate lesion. So it means that EVs are able to induce dose-dependent inflammation of the mammary gland. At the histological context, we also observe an effect of the EVs when we compare to the PBS here. We have very clean tissue, while with EVs we have a destruction of the tissue and a recruitment of polynuclear neutrophils. So we have yeah. And the response here is not equivalent to this one, but we are not so far. So we have also an inflammation in this case, and we measure in the tissue the expression of various markers. And what we can conclude about this is that the response induced by EVs is a little bit different from that of the response induced by the bacteria. Notably, we have a specific chemotaxis-related migratory response in vivo induced by the EVs. The role of these EVs in the pathogenesis is not clear for the moment, but we have a role. So to conclude on this part, what we want to do now is better to characterize the mode of action of the EVs, so the mechanism, the effector, the targets, and so on. The impact probably of small RNA or small regulatory RNA in this interaction with the host cells. We want also to evaluate the role of EVs in bacterial cell-to-cell communication. If EVs can improve the cell response to environmental stimuli, to fasten, for example, their adaptation, to improve the cell fitness, to coordinate bacterial adaptation, activities, and behaviors. And if EVs can decimate specific factors through the bacterial community, for example, something like transient horizontal phenotype acquisition between cells. Okay, so to conclude really this part, not this part, my talk, EVs are really promising tools for many applications in various sectors. But to obtain or to reach this goal, we need some basic knowledge on EVs, notably in terms of their formation, production, the mechanism of selection of their cargo, their mode of action, their activity. We need also we need also to improve our knowledge about their role in bacterial communication, in communication with the microbiota, the host, and the role also of these EVs in the transfer of gene and the evolution of the gene. And finally, I think it's the most important, because we didn't discuss this part today, is the method used to analyze these EVs or to produce the EVs, and we need really to develop a new method for large-scale purification of EVs. So to conclude, I would like to thank you first, but also all my collaborators from the INRARE, and particularly all the PhD students involved in this project, and also Vasco Azevedo and the UFMG for the collaboration. So thank you.
Really nice talk. Congratulations. So about the new mRNAs you find inside the EVs, do you think they can be transcribed inside the host? Or do you find them inside the host?
Brenda, you want to answer the question or not?
Yes.
Vas-y, Brenda. It's your work.
So actually, we don't know if they are found inside the host because we didn't test it. And we don't know if they can be transcribed, but there are some examples in other species that some regulatory RNAs from disease of other bacteria, they can interfere with the host machinery of transcription. So it's possible, but instead of observing, it's not yet described.
You have tried to sequence them somehow?
You mean host cells interact?
No, the
RNA. Yes, we have the sequence of the RNA.
Yeah, we perform the transcriptome of these disease. So we have all the profile of RNAs found inside this area.
So what's the message?
Okay. Just to complete what Brenda said, we have additional result that we are not present today. When we incubate, for example, EVs with host cell, what we observe is an induction of the TLR7 gene. TLR7 is a gene induced by single-strand RNA. And when we block the entry of the EVs inside the cell, with, for example, dinosaur, as I can show you, we have no TLR7 induction. So it means that probably EVs are internalized and liberates their content on RNA, and we have an induction of the TLR7. So I don't know if it's not really the question. The question is the message. The message is that we know that small RNA from bacteria can modify the gene expression of the host by mimicking, for example, mRNA. The big one we don't know. It's small RNA. This is the small RNA. We know that this small RNA can mimic the mRNA or can bind directly mRNA, eukaryotic mRNA, to interfere with their traduction, for example. Or expression.
Ones can be prescribed inside the host and do something?
I don't know. And for the long RNA, and heme messenger RNA, what we think is that it's not probably not involved in with the host, in the interaction with the host, but probably with other bacterial cells. For example, to improve their fitness in providing to the cells mRNA, and the cells are just to translate the mRNA to have the corresponding phenotype. So it's probably like this, but it's an hypothesis.
Thank you.
Yes.
Nice talk, thank you. Is there any uncharacterized proteins inside the vesicles or peptides?
So yes, but it's the same in the genome of almost all bacteria for the moment. At least 30% of the gene, we don't know the function of these genes or the protein coding encoded. So I don't know. I don't know. What we know, the problem with the EVs is that their function is driven by their content. So if we know the protein inside and the function of the protein inside, we can estimate, evaluate their function. If we have a lot of unknown function, we can make a lot of things. Yes.
In the same way of the first question, I was wondering if those long RNAs inside the vesicles, have you ever checked that if they look like a long non-coding RNA or something like that?
So long non-coding RNA does not really exist in bacteria.
Yeah, it has been identified, I think, just in one species. I'm not sure. I don't remember now.
In staff, I don't know. It does not exist in staff. Okay. The long RNA are messenger RNA, and we have operon from up to 10 kb or something like this. But long non-coding RNA, I don't know, because there is no annotation. Because we base our analysis, as mentioned by Brenda, we sequence and we analyze with the annotation. In staff, there is no long non-coding RNA. I cannot answer the question.
I have a question on your internalization experiment that you showed us. You said that after 17 hours, you have still red dots.
Yeah.
Which means vesicles, white. The dye has been diffused, otherwise you will have. So, is it compatible with the role of signaling, for example? Do you think that it could be specific from the cell type, from the environment, something like that? Because for me, it was really surprising that you could see red dots after 17 hours.
It's a good question. It's a good question. Okay. After 70 hours, it seems that the EVs are not localized everywhere inside the cell. It seems that they are in the endosome. Okay. For the moment, this is what we suspect. Why we have no release of their content, we don't know.
And for example, if you perform the same kind of experiment in HT29, for example, you see HT29 with LPS, something like that, you have an effect on the EVs. So
yeah.
I guess that maybe from the outside, you say that
it was out. Yeah.
And a last question, about the scale-up. You were talking about the use of EVs, let's say, at industrial.
Yeah.
So, what do you think that you have to play on what we know already of the biological process? You see, to increase, to push on the biological process, or for example, what we were discussing previously, some physical process. And do you think that EVs produced from bacteria by physical process will have the same functional properties than the...
It depends on the aim of the project. If the project is to deliver something with the EVs, okay. okay, you can use, I don't know, different methods to improve their readings. In our case, what we are interested by how they are formed, by the biogenesis, their content, and so on. So the physical or biological role of this EVs. So you are not interested by artificial EVs. We are interested by EVs produced naturally by the bacteria. So I think that it's not to improve by different methods.
Probably to play on the biological process, and it's more difficult to be able to scale up to industrial level.
Why this exists? No, yeah, exactly. So I was wondering why it exists, because it materials secrete protein for different pathogens. Okay? Would be more interesting, evolutionary, for them to secrete them bacteria from these disease.
Well, now you have an idea about this. Why are you
What was
EVs?
the next question or last question?
I have a lot of follow-up questions, but why these exist? Because the bacteria secrete for different definite way, indefinite way. You will find that when you do the proteome sequencing, the second tome, you will find the protein for the cytoplasm. My point is, for example, it's for protect this protein, it's for deliberate hormone. What does it?
Okay. I've got an answer.
Yeah, but the secret protein, how much way of see there are a lot of secret protein inside of the
Do you know a police, the group?
yeah.
Message in a bottle. Okay. So if you have a message, and if you want to deliver this message at a very distance, what is the best? To put the message on the seat, or put the message in a bottle and then on the seat to protect them? I think that it could be an answer.
Yeah, that's what comes
Moreover, not just one message, but a lot of
That's a lot of message, not one.
So, very nice talk. I'm becoming to be an expert of EV with the thesis of that talk. But I have more translational question because, as you know, in LRA, in Pasco's lab, in my lab, we are working on recombinant lactic acid bacteria to deliver proteins of interest. And you mentioned that EVs could be used for this purpose, and it's a bit difficult for me to evaluate the advantage of using EVs compared to, for example, biocontained recombinant lactic acid bacteria that you will be able and authorized to use. Okay? So this is my question.
Okay, but one advantage is that EVIT is not a living particle. So
for me, it's not an advantage because, in fact, you can
it depends.
an advantage to use a bacterium which will have intrinsic properties, so you can combine both